Okay. Good afternoon, everyone present here. Um, I'm a postdoctoral research associate in Weizmann Institute of Science, Israel. So today I will talk about energy harvesting diverse carotene antennas in proton pump rhodopsins. So first I will give a brief overview about what the rhodopsins are, and then how they utilize these carotenes and as energy harvesting antennas. And then next I'll move to discuss some of our recent findings. Okay. Oh, it's not working. Oh, oh, like this? Okay. So every time like this together. Okay. So first, what are rhodopsins? So rhodopsins are membrane proteins. They are photoreceptor proteins, and they consist 7 transmembrane helix. And inside the proteins, it contains a chromophore, which is a retinal chromophore. This is covalently bound to the protein, and this retinal chromophore actually acts as the light receptor. This absorbs in between 490 to 560 nanometer range. Okay. So rhodopsins can be classified into two classes. One is type 1 rhodopsins, which are microbial rhodopsins. So they are found in bacteria, they are fungi, and they are viruses. And the other one is the animal rhodopsin. It We know this like we have in our eyes, in our retinal of uh, and these animal rhodopsins that we know this acts as a visual rhodopsins. So but in this presentation I'll be talking about microbial rhodopsins. So I'll briefly discuss about the chromophore configuration inside the protein. So this contains the all trans retinal. This is all trans because it so all trans because it has the all the CC double bonds are in trans fashion, and this is in the protein. This is covalently bound to a lysine residues inside and forms a protonated sheaf base. I don't know what. Again, it's not working. It's not moving. Cross cross across. Okay, so next question is uh, so how these rhodopsins act as proton pump, and how they use light as their energy source. So for that purpose, first we need to take a look at the rhodopsin photocycle, and upon absorption of light, this all trans retinal isomerizes to 13 Cs, and then following several intermediates, photo intermediates, it reisomerizes to all trans again. And in one step of these several intermediates, in one step, and a proton is ultimately transferred outside of the membrane. And as you can see that in this outward proton movement process, a proton motive force is generated, and this is used by the ATP synthase enzyme to generate ATP. So this is how this rhodopsins uses light to harvest energy. Okay, so now back to our subject of interest, that does these retinal proteins can contain any other secondary chromophores that can assist in the light harvesting process. So the answer is yes. So the first such kind of rhodopsin was found as xanthorhodopsin, which contains, in addition to retinal, it contains another chromophore, another carotene chromophore named as selenixanthin. And it was found that selenixanthin transfer almost 40% of the excitation energy to the retinal chromophore in order to assist the light harvesting process. So next, following the discovery of xanthorhodopsin, another rhodopsin was found, which is Cleobacter rhodopsin, which has high sequence similarity with xanthorhodopsin. So this was also found to bind selenixanthin, xanthocyanthine, and echinone, these three other carotenes. So the interesting point here is that all these carotenes contains a keto group at 4 positions. So far it was known that only these 4 keto-loaded carotenes can bind the rhodopsins and act as energy harvesting antenna. But like after 15 years, we extended this search process using functional metagenomics and chromophore extraction from the environment. And then we and the idea behind this search was based on to look for some such rhodopsins that has the same fenestration as the xanthorhodopsin has for selenixanthin. And also this glycine was found to be very important for binding of selenixanthin in xanthorhodopsin. So we started our search process based on these two criteria. And the first such rhodopsin was found. So we did a sampling from a lake in Israel, which is a Kinneret lake, and first such sample was found, and we named this rhodopsin as Kinneret 4B8. So with this, first we tested if it binds selenixanthin, and it can employ selenixanthin as energy harvesting antenna. So with the help of absorption and CT spectroscopic studies that can quantify and detect the binding, so we detected that selenixanthin binds the protein, but with the help of fluorescence excitation spectra, which can tell about the energy transfer process. So it tells that selenixanthin fails to transfer any energy. So from this point, we um, moved further with this protein, and then we took another approach, that is from direct chromophore extraction from environment. So for that purpose, we took the protein, and then we did sampling from the same lake, and we took the chromophore extract from the same lake and then mixed together. And after purification of the complex, so this purification, if there is any carotene present in there, that means that this is the actual carotene that binds with the protein. And then with the spectral analysis, we find that indeed there is a presence of some carotene in the in the purified sample, and also CD spectra suggests that there is some species that is present in the sample. And now next, with this purified complex, we moved for HPLC analysis, and this tells that actually we have there uh, two carotenes, which is one is lutein and the other is zeaxanthin. But very interestingly, they are not chelated carotenes as we saw before. And against our like this long belief that only chelated carotenes can bound, so we have here that 3 hydroxylated carotene which actually binds. So now what we did after this from chromophore extraction method, so we moved to test and quantify the binding if the Kinneret 4B8 binds lutein and zeaxanthin. So for that, we bought commercial lutein and zeaxanthin and tested its binding. And as the absorption and CD spectra shows that it has very nice binding. And then when we test the excitation energy transfer with fluorescence excitation spectra, there also we see that up to 50% of the excitation energy is transferred from lutein to the retinal of 4p8. Okay, so next the same we saw for zeaxanthin as well as we saw for lutein. In zeaxanthin also it transfer like 45% of its excitation energy to the retinal. Okay, so um, as I already mentioned that it was proposed that this glycine is very important for binding a carotene in a rhodopsin. So for that purpose, what we did here, we tested the importance of these glycine residues also in our case. And so what we did that we mutated this glycine with bulkier amino acid residues with a phenylalanine. And then what we have, when we test the binding here, we see that in absorption and CT spectra, the binding is abolished. So that means, again tells that glycine is very, very important for the binding of the carotene. So binding of the carotene in this Kinoreth 4B8 as well. Okay, so the next obvious question comes, if there is any functional improvement or any changes of in the functionality of the protein when it binds zeaxanthin. So for that we tested proton pumping in E. coli ferroplast cells expressing Kinoreth 4P8 together with zeaxanthin. And we irradiated the sample with three different light wavelength. One is a violet region, blue and green region. And for control experiment, we also tested Kinoreth 4B8, the glycine mutant system, because this already know that it doesn't bind zeaxanthin. So then we have estimated the rate of the ratio of the rate of proton pumping in between Kinoreth 4B8 and the glycine mutant. And this tells that we indeed have like 33% enhancement under violet light in the proton pumping activity. So zeaxanthin also improves the functionality of Kinoreth 4B8 in terms of proton pumping. Okay, so the overall finding, we can say that the occurrence of carotene antennas in rhodopsins, they are not only limited to the chelated carotenes. Hydroxylated carotenes can also bind rhodopsins. And the bound lutein and zeaxanthin that we already saw, that they can transfer up to like 50% of their excitation energy to the retinal and thus assist in the proton pumping efficiency. And the conserved glycine residues at 153 position here is very, very important for, and it's the determining factor for binding of zeaxanthin. So the open question for us, it remains, and we are already working on it, that what factors actually determines the selectivity of the rhodopsins for their specific carotenes of choice, like to prefer for the ketolylated carotene or hydroxylated carotene, or which position are there, or if they need any combination of keto or hydroxylated carotenes. Okay, so thank you very much for your attention.